Hello, everyone. My name is Maria, and I'm a student at Trinity Western University. And today I'll be presenting my research on the role of hydrochory in Bohemian knotweed seed dispersal and seedling establishment. So, what is Bohemian knotweed? Bohemian knotweed is one of the world's worst invasive weeds and has been frequently observed near water bodies, providing circumstantial evidence for the importance of hydrochory. Bohemian knotweed is of particular interest among other Renutra species as it has vigorous regeneration ability, higher genetic diversity, and is more difficult to control. It also exhibits as parents best properties, Japanese knotweed extensive growth from rhizomes and giant knotweed's viable seeds. Bohemian knotweeds can reproduce both vegetatively, as seen in figure A and C, and sexually, from seen in figure B. Studies on knotweed seeds has been neglected as knotweed mostly spreads vegetatively, and in the past there is not a lot of evidence of knotweed regeneration by seeds. In this experiment, we focus on Bohemian knotweed seeds. Knotweed produces about 50,000 to 150,000 seeds per stem annually. Recent studies have found that Bohemian knotweed seeds actually exhibit buoyancy. Therefore, spread by hydrochory may be a potential vector in long-distance dispersal and providing a method to further invade to new areas. Knotweed species do not have a specific habitat requirement. In its natural habitat in Japan, knotweed was originally found near rivers and was thus promoted to other countries as a plant that actually stabilizes riverbanks. However, it was later found that knotweed actually negatively impacts riverbanks in non-native areas by eliminating ground covers that hold soil during floods, to decreasing river carrying capacity and affecting aquatic species. Additionally, treating knotweed near riverbanks comes with hardships since not many herbicides can be used near water systems, and if they are allowed, they cannot be used within a certain proximity to the water source. Several studies have been conducted on the ability of knotweed seeds and seedlings to remain viable in water for a number of days. For example, Lamberti and colleagues found that seed morphology contributes to seed buoyancy, while Rufida and colleagues found that contact with water favors germination. However, there still exists a knowledge gap on the influence of abiotic factors on water dispersal and thereafter establishment in soil. In this study, the research question was, how does current speed, seed morphology, and seedling growth stage affect the probability of Renutra bohemica downstream establishment? Let's start with the methodology. Achenes were collected in November 2018 to 2020 from 6 different known Bohemian knotweed sites in Fraser Valley, British Columbia, Canada, as seen in the table. The majority of sites were located next to roads and water bodies. The experiment is divided into 2 main experiments. The first experiment is the effect of seed morphology on duration of flotation, germination, and growth rates within various water currents. In this experiment, morphology of 300 seeds, so 20 seeds for each of the 5 sites with 3 current treatments. So the 300 seeds with seed wing intact were measured by taking a photograph of a seed, which was later analyzed on Image J, a software designed to process images for morphology. An example of this is seen on the right figure. Thereafter, seeds were placed into glass bowls, 1 seed per bowl filled halfway with tap water. For this experiment, there are 3 current treatments. The control achenes in no current, C1A, were left in glass bowls of water, but achenes in experimental current treatments were put on laboratory rockers at different dial settings, as seen in the bottom figure. In order to analyze flotation, growth stages, and growth rates, observations were done twice a day until plant mortality. To scale up the laboratory current experiment to better replicate actual stream conditions, a larger container with variable current levels were employed. This time, the current was induced by an aeration pump, and seedlings were grown in Rubbermaid stock tanks, as seen in the right figure. 4 currents along with control were used. The currents were no, low, medium, and high, with current velocities written in the slide. Afterwards, seedlings were grown in water currents until certain growth stages were achieved, either germinating, G1, or exhibiting cotyledons, G2. after which seedlings were transplanted into potting soil and grown for 40 days with observations done twice a week. After the 40 days, each seedlings with root intact were taken out of the soil and their final measurements were analyzed. Final statistical analysis were made on our program, a statistical program, specifically the descriptive statistics, ANOVA, and Tukey tests between sites, current, and growth stages. So let's start with results and discussion. As previously stated, there are two main experiments in the study. We'll first go over the first experiment and its results. So the first experiment was on the effect of seed morphology on duration of flotation, germination, and growth rates within various water currents. This table shows morphological variation between sites. In the first half of the table and the second half, the evaluation of these morphological factors and their effects on flotation in various currents. Overall, this table shows that there is significant difference in seed morphology, yet again displaying how Bohemian knotweed exhibits high genetic diversity, while hydrochory will actually influence the genetic diversity even more and contribute to evolution of renewed species in invaded sites. Regarding the effect of morphology on duration of flotation, it was not significant in no current, but with currents, there were some instances when morphology did play a significant difference on duration of flotation, specifically significant difference in the length and length to width ratio. This table is just a continuation of the previous table. We see again a variation in surface area and surface area to weight ratio of the seeds from different sites. Additionally, that both weight and surface area played a significant difference in duration of flotation in C4 in the high current. Interesting, where in the previous table, we saw that the length and the width played more significant difference in medium current. This table shows that the weight and surface area to weight ratio play a more significant difference in high current as opposed to medium current. Overall, this demonstrates that flotation patterns do get influenced by morphology of the seeds and current of the water body. So throughout the experiment, the germination rates of seeds in various water currents were evaluated. There was significant difference in germination rates between sites, but not between water currents. However, it does seem like the germination rates in water currents are higher than in no current. Overall, this demonstrates yet again the high genetic variation of knotweed strands in different sites, with overall S1 seedlings having the highest germination rates and S4 with the lowest. The presence of current had a significant effect on the number of days it takes for the seed to grow into the desired growth stages. Seeds, regardless of a current presence, would germinate at similar times, but seeds in disturbed water would germinate slightly earlier. The biggest difference is seen when seedlings start exhibiting cotyledons. Seedlings in no current took longer time to grow cotyledons than seedlings in high current. One observation that helps explain this was that seeds in high current got highly disturbed and would often result in a radicle coming out of a seed prematurely or before the mature cotyledons could even grow. In both cases, this would have resulted in seed mortality. A similar pattern is seen with the exhibiting true leaf stage, but as seedlings were more healthier in no currents, they would grow first leaves earlier, which also counts to why they would wither a lot later. Now moving on to the second experiment, which is about the seedling growth in soil post-deposition from various water currents. This slide shows a statistical analysis for all leaf and final measurements. Generally, we see that G2 seedlings, so those planted after exhibiting cotyledons, had the highest final measurement, but G1 seedlings, those planted after germinating, had the highest leaf length and width growth rates, showing that G1 plants generate more leaf growth, while G2 plants generate overall growth. Regarding currents, seedlings in high current had the higher growth rates and final measurements for all factors. Lastly, regarding sites, S1, the Mitchell Library, had the highest growth rates. The significant difference between sites shows yet again the high genetic diversity. However, it is interesting to note that overall seedlings that grew in water prior to soil had a much higher success rate than seedlings grown directly in soil. This figure shows the growth rates of seedlings in soil. Specifically, 3 factors were assessed: current as seen in A, growth stages in B, and sites in C. Figure A depicts growth rates of seedlings that were previously grown under different water currents. The highest growth came from seedlings that were previously grown in high current, and the lowest from seedlings grown in low current. Figure B shows that seeds that were deposited into soil after exhibiting cotyledons had a higher growth rate than those deposited after germinating in water. Finally, figure C shows that knotweed from site 1 from a library had a significantly higher growth rate than other sites. Overall statistical analysis shows significant difference in growth rates between currents, sites, and stages. So in summary, 1, there is significant variation in seed morphology. 2, there is significant difference in germination rates between knotweed sites. 3, seedlings planted after cotyledon stage generate more length and biomass, whereas seedlings planted after germinating generate more leaf growth. 4, the highest growth rates were seen for seedlings previously grown in high water currents. And 5, lastly, there is considerable variation among sites with mission library seedlings exhibiting the greatest success. So results demonstrate that Bohemian knotweed seeds have a high potential in downstream flotation and establishment in soil. This demonstrates that there is a high need in managing knotweed near water systems, especially with the limitations on management given. This study also highlights a great risk associated with increased flooding due to climate change, predicting that with increased flooding, there may be even more invasions by knotweed. So overall, I would like to thank the NSERC and WSSA and Trinity Western University for the funding, the TW Knotweed research team, and Gichel team from Agriculture Canada for help with the research. And thank you everyone for listening and joining in today.